Yes, I think now it's time for Poom Anand to share his presentation. Okay, take your time, Poom Anand.
Nowadays, one problem that farmers are most concerned about is weed problems. And they usually deal with this problem by using a chemical herbicides, which not just cause a pollution into nature, but also can even cause deadly harm to themselves or even us. Good afternoon, our honorable judges. Ladies and gentlemen, my name is Pumanan Cicerin Watts. I'm a presenter of my group, and today I will present you about our science project named Allelopathic extract of Leucaena leucocephala leaves for weed growth and development inhibition. First, let's start out why we're interested in doing this project. Agriculture played one of the most part in GDP, which is up to 8% in 2019, and that's estimately about 50 billion baht. Also can mean that most population in Thailand play a role as farmer or do agriculture. However, they're still using a chemical herbicide to deal with their weed problems without thinking of an adverse effect that may come after it. If we talk about an adverse effect of chemical herbicide or harm, I come up with a case on the man on the bottom right-hand side. His name is Dwayne Lee Johnson. He used to be a school groundskeeper in California, USA. Well, at that time, he lived like a normal person and he really loved his job. But one thing is, he needs to use a chemical herbicide for his job. Unfortunately, one day, his chemical herbicide tank leaks out and then he gets drenched by the chemical herbicides. At that time, he didn't think that this may affect his life that much, so he went to clean up himself pretty neatly and then change his clothes. After that, for a few months, he got rash on his body, so he went to the doctors, and the result come out that he get a terminal cancer. One good news of this story may be he make an impact on using a chemical herbicides, and also he won a lawsuit on chemical herbicide company, which made him a lot of money. But sadly, he will live to use it. And don't be even concerned about ourselves that may risking of having cancers by eating some contaminated fruit or vegetable. Therefore, our team trying to find a solution for these problems. And then we found out one. It's called allochemical. Allochemical is as efficient as a chemical herbicide. However, it does no harm to body. To extend what's allochemical, first let's start with allopathy. Allopathy is a biological phenomenon where one plant inhibits the growth of other plants by releasing allochemicals. So generally, allochemical is just a chemical that the plant releases out to inhibit other plant growth that may compete them for food. So as you can see on the right-hand side pictures, the grass around the tamarind tree doesn't really grow up at all, even though it's out of the shade of the tree. And that means that they get fully right resource and also foods. And according to the research of Siriporn Singsorntiporn, plant in Fabaceae family to have great effect on inhibit other plant growths. Therefore, our team interested in research Leucaena leucocephala leaves, or we commonly call it river tamarind leaves, inhibit effect. It's not just a plant in Fabaceae families, but it also easily grows to our topic area, which means we can easily find it in most part of Thailand. And it have no one use it for really purpose. So we mainly focus on first to find inhibit effects of Leucaena leucocephala and to study effect of an extract at various concentration. Also to test this on our test plant growth and seed germination. For the process, first we start collect our sample of river tamarind leaves at Chok Tree Market, Banpo, Nakhon Ratchasima, Thailand. After we get our leaves, we dehydrate the leaves at 70 degrees Celsius for 7 days to make sure that the moisture does not play any role in our extraction process. Our extraction process, we extract it with 70% ethyl alcohol, which is the best percentage on destroy a bacteria cell. After that, to make our extract as precise as it can be, we filter it with the vacuum filters. And last but not least, we prepare an extract at various concentrations to use on our treatments. First, we have germination test, and next we have growth analysis. Here let's start with first germination test. For the germination test, first we prepare 5 concentrations of an extract, and also we prepare 10 plates. We divide this treatment into 2 sets. First set, we will focus on rice, and the second set we will focus on the spinach. On the first set, we have 5 plates, and on per each plate, we put 50 seeds of rice on it. After that, we drop 5 milliliters of various concentration of an extract, included by first plate will have 0% concentration, a second plate will have 0.1% concentration, third plate will have 1% concentration, fourth plate will have 10% concentration, the last plate will have 100% concentration. In the second set, we do it as same as in the first set, just instead of using rice seeds, we use spinach seeds. We collect the result after 7 days, and to make sure that this treatment had at least error as it can be, we do it for replication. Here for the growth analysis, it's not much different from the spinach, but instead of plate, we use a tube. In our tube, we put 25 milliliters of volume of 0.5% concentration on the tube, of an agar on the tube, sorry. So SMS germination, we have 2 sets. First set, we focus on the rice. A second set, we focus on the spinach. For the first set, we have 5 tubes of rice. We put 3 seeds of rice on per tube, and then we drop various concentration of an extract, as same as in germination test. On the second set, do is as same as in the first set. However, we use a spinach instead of rice. We collect the result after 7 days, and then to make this at least error as can be, we do it for replication. We come to the result. First, let's start with the germination. For the germination, for the rice germination, the treatment here show 3 interesting treatments. First, at 1%, bar chart show that it does have some significantly difference in decreasing of rice germination percentage. Moreover, at 10 and 100%, it does show that an extract does completely inhibits the rice germination. Next, for the spinach germination, it's not much different from the rice. At 1%, bar chart also showing that it does have some significantly difference in decreasing of spinach germination, and also at 10 and 100%, an extract does completely inhibit the germination of spinach. Next, here come for the growth. For the growth, we divide this treatment result into 2 sets. So 2 graphs. For the first graph, we indicate the shoot growth, and the second graph indicates root growth. Here, let's start out with the rice. For the rice shoot growth, the graph showed downward trend, which also showing that the more concentration of an extract show more inhibit effects. And in additional, at 100%, an extract does completely inhibit shoot growth of a rice. Next, the rice root growth. Here, as same as the shoot growth, the concentration at more percent concentration does seem to affect on inhibit effects more than low percent concentration. However, here at 0.1%, it does show some stimulated effects, which is interesting. All right, now it's a graph to compare the shoot growth and the root growth to be more clear for you guys to see it. Here you can see that an extract have more effects on shoot growth compared to root growth. However, the trend go in the same way, and also at 100%, an extract does have base inhibit effects on this presentation. Next, for the spinach shoot growth, the graph also shows downward trend, which also means that higher concentration of an extract does have more inhibit effects. However, here show that an extract seems to have a little bit more inhibit effects on the spinach compared to rice. For sure, at 100%, an extract does completely inhibit the spinach growth, shoot growth also. Here for the spinach root growth, it's the same as the rice, not much difference. However, it's also seen that an extract does have a little bit more inhibit effects on the spinach. As you can see on 10% and 100%, that an extract seems to inhibit the shoot growth of the spinach if compared to the rice. And here this graph also showing that the trend of graph go in the same way, which high concentration of extract does more inhibit effects, and also showing that an extract does have more effects on the spinach compared to rice. So to be summarized, an extract 100% concentration have best inhibitory effects on test plant seed germination and also test plant growth. And interestingly, some percent concentration have stimulatory effects. In development, you may develop this experiment into a real field experiment in terms to see an efficient Allelopathic in field experiments. And also, according to our study, as you can see, an extract does really seem to have great inhibit effects on test plant growth. So you may develop this into business plan into the futures, not just for the farmers to have the herbicides that cause no harm to their body to use, but also for us to not taking a risk of having cancers by eat some contaminated vegetable. Next, you may test this extraction in long periods of time in terms of environmental effects and also the efficiency of Allelopathic. Here's some picture of our working process, and I would like to thank you to these three persons. First, our advisor, Dr. Gmol Chonok, Nาย Next, our lab advisor and also co-advisor, Mr. Ekawat Thara Piksapong. And lastly, our co-advisor, Dr. Thanigan Udom Shloton. Here's some reference, and thank you for your attention. If you have some questions, please don't hesitate to ask me.
Thank you very much, Pumanan, for your presentation, and it's very interesting. And to fulfill the presentation for this topic, I would like to invite Dr. Narisara Pia Sangthong and the Associate Professor Isanan Viwattana Ratanabut to give you the recommendation and ask you some questions.
Ladies first.
Okay.
All right. Good afternoon.
Okay, so good afternoon. I also interested in this field also in case of allelopathy. And I just have a question that you said that you want to control weed, right?
Yeah, that's right.
And weed has a so variety of species, not just only monocot, but also dicotyledon, is it right?
Yeah, that's right.
So what is the main purpose for your research to control which kind of weed, dicot or monocotyledon? Which one? Because you did the experiment in both rice. I think it is representative of monocot, right?
Yeah, that's
and
right.
also spinach, I think dicotyledon. But if you want to control, for example, in case of glass, but it inhibit rice also, how do you deal with this problem?
All right. So as you can see, at 0.1%, it does have some stimulated effects. So you may use the 100% first, and then before you plant the rice, you can use you can like plant water the field, and then after that, the concentration of the extract may be lower, and then you can plant the rice. And also the half-life of chemical, this allochemical, which is levodopa, is have just 1.5 hours for half-life, so it's really easy to degrade.
Did you try already to check it?
No, I haven't tried on field experiment, but just according to the research.
Yeah. I see. Because if you plant, for example, if you put in the real field, I think you need to get the soil sample and then analyze it. That still have some extract in the soil or not before you plant the rice. Is it right?
Yeah. So that's maybe developed in the years.
I see. Okay. Thank you.
All right. Any other question?
Okay. Pumanan, I'd like to tell you that your presentation is very good for me. Okay, in my opinion, your speech and your pronunciation mostly is very, very good. You are one of the good students. And this project is also very interesting for me too. But I would like to ask you one question. Why did you choose those 5 concentrations for the study? In slide 15.
May I share screen again?
Okay, why did you choose those 5 concentrations in slide 15 for this study, for your study?
Yeah. Pardon me, sirs. Could you say that question more clearly again?
Not 15, maybe 16.
Oh, 16.
maybe, yeah, 16, yeah.
Yeah.
Why did you choose these 5 concentrations of extract?
Oh, so 5 concentration to show the variety of the concentration of an extract. So you can see an effect of an extract at various concentration, not just 100% concentration, because it may affect differently and when it dilutes or something, so you can be develop this into the future more easily, sir.
Yeah, but I mean, by for examples, maybe you can choose 0%, 20%, 40%, 60%, something like that. So I just asked you why you choose 0.1, 1, 10, or 100%.
All right. So according to my study, most of the research use 10 times better than the lower concentration to see the best various. I think the best various effects on at various concentration of an extract. Yeah. So according to the research that I study on, they mostly use this. So I prefer to use this.
Okay. So you prefer to use concentrations.
Yeah, I prefer to make it 10 times more better. Yeah.
Okay.
All right. Any other question?
Okay.
Yes, I have another questions. I just want to ask that why did this extract effect on shoot growth more than root growth? Because you show that it inhibit shoot more, right?
Yeah, that's right.
Is this correct? Yeah. Do you know why?
So this interestingly, and I'm not sure if this answer will be right answers. However, according to our research, it seems that an extract have effects on the shoot more than the root. Maybe because the roots will go out before the root come, right? So it may inhibit based on both root and shoot. However, it will inhibit the root. When it inhibits the root first, the shoot won't grow. So that may be a reason why.
I think it's possible. Yeah.
Yeah. Any other question?
I'm not sure
Okay.
how about the time. Is already?
Oh, yeah.
Yeah, the time is up. Thank you
So
very
thank you
much
so
for
much.
the presenter Pumanan, and thank you very much for the distinguished committee. Okay. And
Thank you for your nice presentation.
Thank you so much.
okay, before we start the next presentation, I would like to ask the committee to submit the evaluation form for the first topic in this afternoon. And for the next presentation is the genetic modification of Saccharomyces cerevisiae to directly hydrolyze starch. The presenter is Mr. Pitayut King-Gao. Are you here, Pitayut?
Yes, we are here.
Yeah, good afternoon, Pitayut.
Good afternoon.
Okay. Yeah, I think you can do it very, very, very well this afternoon. So before you start the presentation, could you please double check for the screen sharing?
Okay.
Yes.
Do you see my slide? Where?
I just see the square. I don't see the full screen.
Where?
Yes, I think it's okay now. Okay.
All right.
Yeah. Before you start to present your work, I would like to remind you about the time. When 10 minutes pass, I will ring the bell like this. And when the time is up, I will ring 3 times. Okay?
Okay.
Okay. Just do your best. Please welcome.
Good afternoon, professors. My name is Pitayut Kingkao. This is my great honor to present my project, Genetic Modification of Saccharomyces cerevisiae HC90 to directly hydrolyze starch. This is our outline. We have introduction, methodology, results of the experiments, and conclusion. Let's begin with introductions. Energy. Energy is a fundamental ingredient in human life. There is no industrial, agricultural, health, domestic, or any other sorts of process that doesn't require energy. At the present day, we cannot abandon any existing energy sources. They must reduce the environmental impact, and new sources must be added, especially alternative ones. And ethanol is one of the most important alternative energies. Ethanol production industry has been an important source of energy to replace imported fuels from other countries. In Thailand, cassava and molasses have been used as the main materials to produce ethanol. Traditionally, bioethanol fermentations from starch involve 2 steps. The first step is the digestion of starch into sugar by using alpha amylase enzyme and glucoamylase enzyme. And the second step is the fermentation of sugar into ethanol by using yeast Saccharomyces cerevisiae HC90. The 2 steps need at least 72 hours. They are time-consuming, high cost, and high energy. To save more time, more money, and more energy, we aim to combine the 2 steps into one. We found that the first step requires enzyme alpha amylase and glucoamylase, and the second step requires yeast Saccharomyces cerevisiae HC90. Is it better, right, if we can use yeast that can produce enzyme? In this study, we use CRISPR-Cas9 technique to genetic modify by inserting a mu3 gene which expresses alpha amylase enzyme and GAI gene which expresses glucoamylase enzyme. The genetically engineered yeast is therefore now able to use starch as substrate. Next is methodology part. Overall, we have 4 chapters. First, genetically engineer yeast cells to transfer mu3 and GAI genes. Second, DNA extractions to get DNA for PCR. Third, PCR checks to confirm that the 2 genes were integrated into the yeast genome. And the last one is starch hydrolysis to detect the function of the genes transfer. In the first chapter, genetic engineered yeast cells, competent yeast cells were prepared with donor vector and guide RNA, then electroporate DNA into the competent yeast cells by electroporation, and then plate all the cells on YPD plus antibiotic plate. Next is DNA extraction. The grown cells was mixed with lithium acetate plus 1% SDS, then incubate at 65 degrees Celsius for 5 minutes. 95% ethanol and 70% ethanol were added to clean cells respectively and remove and dissolve in TE buffer. Next is PCR check. The PCR check was used to check the presence of the 2 genes that were transferred into the yeast genome. And then the PCR product were used in the gel electrophoresis processes. And the last process is starch hydrolysis. The engineered yeast cells were grown on starch plates, then flooded with 2% iodine solutions. And the clear zone that occur around the colony will be observed later. Next is results of the experiment. The first result is from PCR check. The transformed yeast were checked for the presence of the gene that integrated into the yeast genome by PCR. Gene-specific primer pair were used for the detection of MU3 and GAI gene. The double insertion were also checked with each specific primer pair separately. The figure 1 shows all the yeasts that transfer MU3 gene have MU3 genes, and figure 2 shows all yeast transfer GAI gene have GAI genes. Figure 3 and figure 4 shows that the double insertion, some of them have MU3 genes, but all of them have GAI genes. Next is results from iodine test. The enzyme activity can be detected by iodine test. The selected yeast all contain activity of alpha amylase enzyme and glucoamylase enzyme. From the figure, the first group, yeast transfer MU3 genes, found that A4 clones has the most average clear zone. In the second group, yeast transfer GI genes found that G6 and G9 has the most average clear zone. And the third group, the double insertions, found that GA21 clones have the most average clear zone. Overall, the double insertion have the most average activity of the both enzyme, following by MU3 gene and GI gene. Next is conclusion, but before the reach of conclusion, I want to talk about overall from the results of the experiments. From PCR checks, the result shows that genetically engineered yeast found that the existence of the MU3 and GI genes. And from iodine test, we can detect the function of the genes transfer in the yeast by testing the change into sugar by iodine solutions, and the colony, the clear zone around the colony were observed. We can conclude that CRISPR-Cas9 technique can be used to transfer the genes into the yeast genome, and a genetically engineered yeast can express enzyme that can hydrolyze starch. We hope that our engineered yeast can be applied in ethanol production industry in the future, and this is the promising start for future ethanol producing yeast development. Any questions?
Thank you very much for your presentation. And now it's time for Q&A. Please welcome Dr. Narisara and Dr. Isanan.
Dr. Nursa, would you like to ask first?
Now I'm thinking about the question. But first of all, I just want to say that I just want to ask one thing, that you did this diagram by yourself, the drawing.
Which diagram?
Yeah, in your slide, it's like ethanol production process or something that show, because it's very nice. It looks like if you draw it, it's very easy to understand.
Excuse me. Actually, I have something similar questions.
This slides.
Yeah, this one is so nice, and it's easy to understand the step from cassava to ethanol. Did you make it by yourself?
Yes, but we modify from the internet.
Yeah, but it's very nice.
We did it by ourselves.
Oh,
Okay. So, excuse me. Actually, my first question is similar to you, Dr. Narisara's question. But I would like to ask you, did you did all experiments that you present today by yourself?
Yes, we did it by yourselves because our professor very strict with our project. She just guide us what we should do, but she didn't help us to do them, because we should learn by ourselves to understand truly in our project.
And also you prepare all slides presentation today by yourself?
Yes, we do all the presentation.
Oh, excellent. Excellent. Very good.
Thank
Very
you.
good. And if so you also prepare competent yeast cell by yourself?
Yes. Our advisor just guide us like we should do like this, guide us the process. But when we use the equipment, we should do by ourselves.
Okay. No more question for me.
I see. I think that you want to reduce the step to make ethanol, right? From
Yes.
from 2 steps, because normally it will start from starch and then get glucose,
Yeah.
right? Yeah, and then
Start
from
glucose
sugar,
and ethanol.
yeah, to ethanol. As we know that yeast like produce some enzyme, maybe succinate dehydrogenase to like make a sugar become ethanol. Is it right?
Yes.
And how long does it take? If you want to compare, if you want to use this yeast for in case of commercial something like this, how long does it take? If you compare with like if you need to use 2 steps as the previous thing that they do, but if you use your yeast,
Like count the time, like count the time, how long that our engineer yeast work. But I am surely that it can reduce time because it's just one step.
I think this is very interesting research because if you can reduce the time from like 72 hours is about 3 days, right? And
Yes.
if your yeast can reduce time also, it's just only 1 step and you do not need like so long time and then make ethanol. So it's very nice.
Thank
Yeah.
you.
Do you have any other plan to do for the future work?
We are doing DNS assay to detect the amount occur in the activity, like how many sugar that the yeast can produce, can hydrolyze from starch by DNS assay.
Yeah. And then go to the another step to
To
maybe.
apply in the real ethanol industry.
I see. Yeah. For me, I think no more questions. Let me ask one more question. Is it okay?
Yes.
Yes, you
So how
can.
about double insertion? Why it showed the result that show is better than other 2 conditions?
Uh, the uh, the two enzyme like can have the best activity when they work together.
When they work
Like they can
together.
cut like the bond between glucose more than just one enzyme.
So that means if you like in the activity, if 2 enzymes used together, you can get a better result. Is it right? It's like synergistic or something. Hello. Maybe freeze, right? เหมือน
Yes.
น้องยังอยู่ไหมคะ
เกิดอะไรขึ้นเหมือนแห้งนะ
ของเราไม่มีแล้วนะคะ For me no more questions but I think maybe something problem with the connection, right?
Yeah, it might. Yeah, I think it like there is some trouble from the internet connection from Pitayut. Yeah, but it's okay now. Thank you very much for the presenter Pitayut Ginggao. And for the next presentation is the study of the ability of climbing plants to entrap dust. The presenter is Miss Thamaporn Pansanit. Miss Thamaporn, please show you, or the technician, please. Oh, okay, thank you. The technician will add Thamaporn to the meeting room. We have 3 more presentation in this afternoon. We will start, after the third presentation from Thamaporn finish, we will have the last break for 2 more presentation: monitoring of ambient nitrogen dioxide concentration in Mueang Trang district by passive sampling method, and low cost developing bioplastic film by using tamarind seeds and polylactic acid. And we will have other 2 distinguished committee: Assistant Professor Kornkamol Suwan and the Associate Professor Thavitam Limpanupa for the last 2 topics. Okay, and now I think Thamaporn is here. Good afternoon, Thamaporn.
Good afternoon.
Okay, are you ready?
Yes.
Okay, do your best. And before start your presentation, please double check for the internet connection and check
Okay.
your microphone, and please prompt with your screen sharing.
Excuse me, how to share, because I'm first time to do in iPad. Because
Okay.
my notebook don't have a camera.
Okay. I'm not sure the technician said that you cannot share your screen from the iPad. You can just only use the laptop for sharing your screen.
Okay, so
Or
but
yeah.
maybe it don't have my face, and I'm not sure about the sound, because just like the electric have shut down this afternoon and everything is gone.
It's okay to switch to use your laptop now, and after
Okay.
the presentation, you can show your face again by using your iPad.
Okay, I will try.
Is it okay?
Okay, so just enter in the computer. Can you see me?
Okay. Just a moment, please. I will ask the technician to fix this problem. เจ้าเจ้าหน้าที่คะอันนี้น้องสามารถใช้2อุปกรณ์ในการนำเสนอได้ไหมคะคือสามารถล็อกอินเข้ามา2อุปกรณ์ได้ไหมคะเพราะว่าโอเคค่ะได้นะคะ Okay, you can use both your laptop without the camera, and you can show your face using your iPad. But you have to turn off the microphone from one device. You can use just only one device for the microphone.
Hear me now? You see me or not?
Yes, I see you.
So you see my skin or not? I'm just share my skin.
Please turn off your speaker and microphone one device. Yes. And if you'd like to show your face, could you please turn your iPad? Okay, yes.
โทษครับน้องครับปิดไมค์คอมแล้วก็เปิดไมค์ iPad ดีกว่าเนาะ
อัน
ปิด
นี้
ไมค์
พี่
ที่
ได้
คอ
ยิ
ม
นไหม
นะครับ
คะ
ได้ยินครับ แต่ว่าปิดไมค์ที่คอมได้ไหม
โอเคค่ะ
โอเคครับ
ตอนนี้ปิดไมค์ที่
ตอ
iPad
น
แล้วนะคะ ปิดไมค์ที่
ตอน
คอม
นี้
แล้วนะ
ที่
คะ
ตอนนี้ที่ตัวคอมพิวเตอร์คือสามารถเข้ามาจอยใน Meet ได้ยัง
ใช่
คะใน
ค่ะ แต่ว่า
Meeting
ได้แล้วค่ะ แต่ว่าพอแชร์สกรีนมันแชร์ไม่ได้อะค่ะ
อ๋อ ตอนนี้น่าจะต้องให้ทางเจ้าหน้าที่ช่วยแอดน้องเข้ามาอีก account หนึ่งไหมคะ หรือว่า1 account สามารถ sign in ได้2อุปกรณ์ไหมเอ่ย
เดี๋ยวหนูให้เพื่อนลองแชร์สกรีนให้แทนได้ไหมคะ หรือว่ายังไง?
โอเค รบกวนน้องรบกวนใช้แชร์ PowerPoint อะค่ะ เปิดที่ PowerPoint เลยค่ะ ตอนนี้เหมือนหน้าจอสกรีนมันมาแล้วค่ะ
อ๋อ โอเคค่ะ ได้ค่ะ
ค่ะ
อันนี้เห็นยังคะ?
เห็นแล้วค่ะ รบกวนเปิด PowerPoint เลยค่ะ แล้วก็รบกวนปิดลำโพงที่คอมพิวเตอร์ แล้วก็ปิดไมค์ที่คอมพิวเตอร์นะคะ ลดเสียงลงให้สุดเลยค่ะ เพราะว่าถ้าไม่งั้นเดี๋ยวจะเป็นเสียงมันจะสะท้อนนะคะ
โอเคค่ะ ตอนนี้เห็นหน้าจอคอมแล้วใช่ไหมคะ?
เห็นหน้าจอคอมที่แสดงตรง This PC แล้วก็มันโชว์หน้าตาของไดรฟ์
ตอนนี้หนูเปิดเป็นอันนั้นแล้วอะค่ะ เปิดเป็น PowerPoint แล้วอะค่ะ
ไม่เห็น
ไม่ทราบว่ามัน
ไม่เห็น
มัน
PowerPoint
ทำ
อะค่ะ
ยังไง?
ไม่เห็น PowerPoint เลยค่ะ เดี๋ยวลองออกแล้วเข้าใหม่นะคะ
ได้ค่ะ
ค่ะ ค่ะ เนื่องจาก topic นี้ก็คือจะเป็นของน้องคนสุดท้ายสำหรับกรรมการ2ท่าน ก็คือท่านดร.นริศรา กับท่านรองศาสตราจารย์ดร.อิสนันท์นะคะ เพราะฉะนั้นก็จะขออนุญาตใช้เวลารออีกสักนิดหนึ่งนะคะ ตอนนี้ก็คือตามตารางเวลาเนี่ย ก็คือของน้องจะเป็นบ่าย 2:50 นาทีนะคะ ตอนนี้เนื่องจากมีเหตุขัดข้อง